acid is present. So, and this inactivates the C3b. Now factor B, C3b on the surface of a foreign cell, another plasma called factor B. Now factor D, what happens is when C3 and C3b and C3a are completely present together, what will happen is you have binding of C3b to factor B. So this will allow the factor D to be cleaved from the factor B to form BA and BB. So B is your inactivated form, capital. Whenever you have the small letters, it is the activated forms. So factor BB is an activated form which remains bound to C3b. So you have C3, capital B, small b, which is known as your C3 convertase, along with factor D, which is the properdin. So properdin, also called as the factor P, now is going to bind with this complete C3BB complex. This whole thing will be stabilized. So this is your C3 activation complex. This again helps in production of more amount of C3b. And finally, all this is going to be repeated and it is taking place in a time of around 5 minutes. Then when this activation complex comes into place, it leaves the C5 into C5a and C5b. And again, you have the production of membrane attack complex. So final overview of alternate pathways, C3 to C3b, then C3bb is formed finally, properdin, and then this is known as your C5 convertase. Finally, this C5 to C5b plus C5a forms your membrane attack complex. Now we have the third pathway. This is the lectin pathway. Originates with the host proteins, binds to the microbial surfaces. So we have the mannose binding lectin. So like it suggests, lectin is a protein that is going to bind to carbohydrates. What is the carbohydrate over here? It is mannose. So what is going to happen is this mannose residues are present on your glycoproteins or glycolipids, and these carbohydrates that are present on the microorganism surface. So because of which whenever lectin goes and binds to the mannose, you have the mannose associated proteins like serine proteases 1 and 2, which are structurally similar to that. Again, induces inflammatory responses. And then MBL associated serine proteases to MBL. So this mimics the activity of C1R and C1S, and this causes cleavage and the activation of C4 and C2. Again, this is antibody independent, just like your alternate pathway. Except for the C1 proteins, all of it. So this is how your membrane attack complex is formed with all the three classical pathway, the alternate pathway and the lectin pathway coming together. And finally, you're going to have a membrane attack complex that is formed. So how is it going to be regulated? You have a C1 inhibitor, which is an important regulator of classic pathway, binds to serpin, which is a protease inhibitor. So it is one factor, and then you have something called as the factor H, which is going to regulate your the alternate pathway. So what is going to happen is amount of C5 convertase, what is necessary for the formation of membrane attack complex, is going to be stopped or reduced. So this is one activity that is done. And then you have formation of properdin also, which is going to be regulatory in nature. So protects the C3b and stabilizes the C3 convertase. Now we have the cleavage factor, that is C3b and C4b, which inactivates the active forms. And then we have something called as the decay accelerating factor, which helps in, which is a basic glycoprotein which is present on the cells. So what happens is, because of the presence of that, it will prevent the assembly of this particular complex. C3 convertase complex or C3BB complex is not formed. So because of this, what is not formed finally? Your membrane attack complex. So this is going to act on both your classical as well as. Of course, we have a C4B binding protein also, and it's a cofactor for factor 1. And you have complement receptor 1s and protectants and nutrients that are present. So all of these are your regulatory aspects or factors in your basic complement system. So I have finished speaking about the complement system. Now I want to talk about the immune system that is present in the health and disease. So here we are going to see the basics of your hypersensitivity that is present. So what exactly is the meaning of hypersensitivity? Something which we do not want in our body. In the most easiest way to tell. Why? Because it produces an immune response which is not desirable. Only if there is an infection, you want an immune response. But otherwise, I do not want an immune response triggered in the body. But then this is happening. So when an immune response results in an exaggerated, so maybe say for example, you need only one molecule of antibody, but then 100 molecules of antibodies are formed. So it is an inappropriate reaction, which is not necessary. So this causes harm to the host. So that is when you term the use the word hypersensitivity or allergy. There again, based on the kind of mechanism that is involved, it is of 4 times. What is the time taken is also important to know what kind of hypersensitivity is and what is the type of disease that is involved. So you have type 1, 2, 3 and 4, and or in type 1, 2 and 3 you have antibodies that are being released, but type 4 is only using cell mediated hypersensitivity. We will just have these. So when you classify them, type 1 is when you have your immunoglobulin E, rarely G4, which is being released. What is the kind of reaction that you get? Immediate reaction. Type 2, IgG and IgM, and it is cytotoxic in nature. And type 3 of hypersensitivities, you have antigen-antibody complexes formed. So it's an immune complex reaction. And type 4 is when your T cells are released and the cell-mediated. So type 1 is commonly called as allergy. It is mediated by IgE antibodies that are going to be produced by your B cells, plasma cells. Why is it going to be released? Because T helper 2 types of cells are going to be released, because of which whenever it is going to take place in the presence of antigens. So these antigens are called as allergens. What are they? House dust, pollen, different kinds of caustics sometimes, and certain insects, mites, dust mites, which mites cause, and certain kind of very rough clothing or very irritated clothing or certain drugs. So some people are allergic to penicillin. So it says that it is an allergen that has come into place and it starts releasing your antibodies. How and what? It is going to release IgE antibodies because Th2 cells have been stimulated. Exposure can be through ingestion. So when you take a drug, or inhalation of the spores, pollen grains, or through an injection or through direct contact. So type 1 hypersensitivity can be systemic, that is anaphylaxis as it is known as, complete body is involved, or localized in one particular area. Example, allergic rhinitis or asthma. Allergic rhinitis, you start having symptoms of a cold. Asthma is your breathing problem that you get to see. Type 2 type of hypersensitivity, you have IgG and IgM antibodies that are released. They go and act on the cell surface antigens. Then what will happen? The complement system is going to be triggered. So the cell, again, your membrane attack complex is going to be formed and damage to that particular cell. So example of this kind is your transfusion, blood transfusion, wrong from A to B when you're giving. It causes your type 2 hypersensitivity. and HDNB, your hemolytic disease of the newborn, Rh factor negative positive will cause you type 2 hypersensitivity. Then type 3 hypersensitivity, what is going to happen is formation of an immune complex is taking place. So you have the cells, you have different kinds of antigens. Finally, there's a complex that is formed. So it'll take quite some time, in fact, 3 to 10 hours after the antigen comes out. Example is your arthros reaction, and example is serum sickness. It can be both internal or external, exogenous or endogenous. What is happening is antigens are there inside the body itself, and sometimes wrong identification of the antigen, like that a particular component as an antigen will trigger the immune response. So you have a complex form wherever in the body they are deposited and they activate the complement system again. And then PMN cells are attracted, and because of which there is a lot of inflammation and tissue injury. So antigens will form with the antibodies. You have an antibody complex, and phagocytosis will come and try to remove it. Activation of the complement system is taking place. Inactive complement is also there, activated complement, and the whole thing will start triggering, or neutrophils come into place. There's this inflammatory reaction taking place. So because of the inflammation, whenever there is an inflammation, there is a damage to the blood vessel. So this is how type 3 type of hypersensitivity takes place. Example is in the case of systemic, wherein the whole body is affected. You have immune complexes. You will see that particular reaction in say a few days to 2 weeks almost. Say example foreign drugs or whatever is being given. So what is going to happen? The side effects of it are going to be a fever, urticaria, that is hives, arthralgia, meaning your system is going to be inflamed, eosinophilia, eosinophils are going to be increase in the size of the spleen, splenomegaly, and lymphadenopathy, or like inflammation of the lymph vessels or lymph nodes. So this is serum sickness. In Arthur's reaction, since it was Dr. Arthur's who had actually discovered this, so they seen that it is there in the local area itself. This kind of inflammation. So it is known as hypersensitivity pneumonitis. Your systemic lupus erythematosus, your rheumatoid arthritis are all types of or examples of your type 3 hypersensitivity. Now we have certain immune complex disorders. We will just have a look at it. So hypersensitivity, pneumonitis, glomerulonephritis, arthritis, systemic lupus, like I told you. So all of this, what is happening is antigen will produce antibodies, activates the complements. Glomerulonephritis. So these on your kidneys, on the kidney glomerulus or glomeruli, the cells of the kidney, you have immune complex deposition. So this will cause kidney failure. Rheumatoid arthritis, inflammation on the joints. So again, basic symptomatic relief is what is necessary. This is how it becomes because of rheumatoid arthritis. Systemic lupus erythematosus, autoantibodies against the DNA itself. So against your RBCs, platelets, lymphocytes, muscles, all of it is formed. So what can be done? You will only treat the symptomatic part because there's nothing else that can be done. So the butterfly, as it is known as, this is how it will become basically in the case of systemic lupus erythematosus. Now let us look into hypersensitivity type 4. In this, what is happening is it is also known as cell-mediated hypersensitivity or delayed hypersensitivity, wherein you have T lymphocytes that are going to be reacting. So in this, you have, after the antigen comes in touch with the particular wherever exposure in start after hours, and it will go on to days. So what can be happened? So it can be transferred by sensitized T cells, but not through the serum as such. So different kinds of virus, fungi, parasites cause this type of hypersensitivity. So because of release of macrophages and cytokines. So what happens? First exposure, you have APCs that come, macrophages, and then delayed T helper cells and T helper type 1 cells. Second, you have production of interneurons. And then again, activated macrophages will be there. Finally, it is going to be killed. So different kinds of hypersensitivity reactions which are important are tuberculin response. So whenever tuberculin, which is an injection, is given, so that if the person has tuberculosis, it is going to show a reaction. So this is how you use type 4 hypersensitivity for your own advantage. That is for disease diagnosis. Example, tuberculosis. If an individual is not affected or not has not been vaccinated, you will not have a response. So you'll have just a red heart swelling that is going to be developed in the people who have been exposed or infected or immunized against the tuberculosis. So we have BCG vaccine, because of which whenever we are taken, this can be seen. But depending upon the active infection and passive infection, we will know whether only vaccination has caused this or whether there is any kind of infection. And then we have allergic contact dermatitis, wherein you have a cell-mediated response. So you have a very irritable skin rash. So it can be any kind of modified skin proteins that are going to be treated as foreign, and you're going to get some blisters and different kinds. So finally, what is going to happen is you will have a steroid. So this is the kind of oozing also that you can see, blisters coming up with fluid that is going to come out of the skin, especially in the case of allergic contact dermatitis. And we have graft rejection. So whenever there is any kind of organ tissue transplantation, that is known as grafting. So they will recognize, or the body will recognize the donated liver or kidney as foreign and start producing antibodies against it. So you have major histocompatibility complexes also acting on the graft cells. So this can be done by giving immunosuppressants. So what are the different types of grafts that are seen? Right. So you have different types of grafts, which is known as isografts, allografts, and xenografts. So you have a genetically identical sibling clone from the body itself. We are taking that part and putting. Allograft is different member species you are grafting it. Xenograft is from from the rat to the cat. So it's a xenograft. So that's one thing. And the whole of hypersensitivity reactions have been given in the in this particular table. You can just see type 1, type 2, type 3, and type 4. What are the kind of reactions that are taking place? First is IgE, IgG, and M. Ig, what do you call it? You have complexes, immune complexes that are being formed over here. And then you have the cell-mediated hypersensitivity and the different kinds. So this type 2 is also known as complement activation or antibody-dependent cell cytotoxicity. So, and then when it comes to the references, has also been given here. You can just have a look at it. Now I want to start with the next topic, that's the vaccines. So what exactly are vaccines? What are the types? What are the uses? We just have a brief introduction. So these vaccines are going to improve your immunity to a particular disease. So it contains an agent that resembles a disease-causing microorganism. So it'll be that can be a weakened form or killed form of the microbe itself, so that it will not cause the same disease, but it will trigger the immune reaction against that particular disease. So the agent stimulates the immune system and then keeps a record. Your memory cells are going to do that. So term vaccine has been derived from variole vaccinae, which is the smallpox of cow. So we know that it was Edward Jenner who had used it to denote the cowpox. In fact, so what did Edward Jenner do? He was working as a surgeon apprentice under the surgeon who was working as an apprentice, that is an assistant. So he saw that whoever had were exposed to cowpox, then they did not have smallpox, which was fatal. They only had a mild dose of it, and it was not fatal, or it was not as disfiguring as it was for others. So he wanted to know why. So whoever had cowpox, he took that particular pus from the cowpox and put it into an 8-year-old boy. Then 6 weeks later, he took the smallpox vaccine or smallpox scraping, and he injected into the boy. He saw that so finally he saw that it was safe for both children and adults. So that is how vaccine was developed. And then in 1880s, Louis Pasteur developed vaccine for chicken cholera as well as anthrax. So by the time it was 19th century, vaccines were very famous. So what are the different types of vaccines that we look at? So we have something called as live attenuated vaccine. So what is it? It is a live microorganism that is there. Attenuated, meaning it is weakened. So it does not have the property to cause disease, but at the same time it is live. So these are teachers of when it goes into the immune system, it is going to trigger a response. Example is vaccines against measles, mumps, chickenpox. Second is inactivated. So what are they doing? They are going to kill the organism, but the protein that is going to be there, it's going to be taken. Such vaccines are stable and they are safer than the live vaccines, which can cause disease at any point of time. Example is flu, polio, and hepatitis A, rabies vaccines. You have something called subunit vaccine. So, entire microbe, only the antigen is taken. This is going to stimulate the immune system. So your T cells will recognize. So example is your plague immunization. And then you have toxoids. So that means you're not even taking the live or heat killed, or you're not taking any part of the organism as such, but you're taking the toxin that is being released by the microorganism. And these vaccines are then used when the bacterial toxin is going to cause the illness. So you can actually inactivate the toxin by you treating it with formaldehyde or formalin. So these are known as toxoids. Example, Crotalus atrox toxoid, which is a vaccination against rattlesnake bites that is given to dogs. Tetanus toxoid. Your TT injection is also a toxoid only. Okay, now you have something known as the conjugate vaccines. In this, if a bacterium has an outer coat of polysaccharide, that is the polysaccharide layer that is present as different kinds of bacteria do, then you can make a vaccine out of it. So you can take a polysaccharide coating, then disguise this bacteria's antigen against it, so that your immature immune system, whatever is there, will be given, and then it can be used to trigger the memory cells. Memory cells will give example this your Haemophilus influenzae type. Then we have some known as DNA vaccines. So what is happening is this: your DNA is taken, and then the whole organism is incorporated with it, and then you get it back into your system. So what will happen is this is going to trigger your immune system again. Example is influenza vaccine. Now we have recombinant vector vaccines, again experimental. So wherein attenuated, that is your weakened, or the bacterium is going to be introduced along with the microbial DNA into the body cells. So you have a vector, that is a vehicle that is being used over here. It can be a virus or a bacterium. And example is your DBT vaccine. Something known as adjuvants. Common alum is an adjuvant. Vaccine is having an adjuvant. What is it? This adjuvant is a substance which, when attached or added along with the vaccine, increases the body's immune response against the vaccine. So example is saponins, common alums. They're all natural glycosides. So they're all carbohydrates that are present to the steroidal nature or triterpene. So they have different kinds of pharmacological as well as biological activities that are together. And they can act as adjuvants. Most commonly used are Quil A, derivatives of QH21. You can use alamos also, like I have already told you. So what can be done is isolation of Quillaja saponaria is taken and evaluated along, the amount of adjuvant that has to be given is evaluated there. So because of the presence of adjuvant, both TH1 and production of cytotoxic T lymphocytes is going to take place, making it a very effective response against that particular vaccine. So it is used as therapeutic cancer vaccine also. So World Health Organization is having a separate R&D unit only for vaccine developments. So still HIV vaccine is one of the top priority. But right now, we can say that, it has shifted from HIV to COVID, unfortunately. But then that is how it has taken. So you have your Vaccine Research Center and Division of Acquired Immunodeficiency Syndrome, that is AIDS, NIAID as it is known as. So we are trying to find out a vaccine to AIDS even today. So that is going on. But of course, we have found out different kinds of COVID vaccines, though it is not effective. As such, you need to keep giving it doses so that, even if you are affected with COVID also, mild infection occurs. So this is all about vaccines. Now I want to talk about the basics of animal biotechnology. Let us just see, there is 15 minutes more. So give me a moment. Pause my share. So now we have scope and concepts of animal biotechnology. I will just start resuming share. So what is biotechnology? We need not see, we have this everywhere. So manipulation of the living organisms and different kinds of organic materials to serve the human needs. Example is like since time ages on, we have used the concept of curdling of milk. We use organisms, that is the starter culture. You just take into a cup of milk, you add a little bit, 1 spoon of curds, which is a starter culture, to it, so that this whole cup of milk will turn into curds. So that is inadvertently, without us knowing also, we have been using methods of biotechnological methods, in fact. And at the same time, ages ago itself in the, in Egypt, yeast was used for making breads, for making alcohol. And we are using different kinds of beneficial organisms so that, which will kill other organisms as such. Your antibiotics, your cloning of plants or artificial insemination, all this is basically biotechnology in scope and applications. So application of biological organism, system or processes to manufacturing and service industries. So live organisms, dead organisms, their products, whatever, and their components is used where in agriculture, food, industry, all these processes. So finally, you can say application of scientific and engineering principles of the processing materials by biological agents to produce or provide goods and services technically. So we see that that's what fermentation, production of wine is one of the most oldest, and then your production of curds, then dosa idli batter also is being fermented over here when we come to India and we talk about this. So all this is like you're using the principle of fermentation without even knowing that yes, you are being using biotechnology. So you can see that it has, and today you can see one of the greatest advancements are there. You have your PCR, RT-PCR, and different kinds of DNA technology, DNA fingerprinting, DNA cloning, whatever you want, it is there in biotechnology. So when it comes to the history, you can see that in 6000 BC, so Sumerians and Babylonians, then Egypt, they were able to produce alcohol. And then 2500 BC, this is much earlier, so Aryans started preparing curds, and Greeks were one of the people to start making cheese. And 1822 to 1895, Louis Pasteur is also known as the father of microbiology, father of industrial biotechnology, discovered that yeast is going to produce alcohol in wine, and these rod-shaped bacillus basically produced acetic acid and souring of wine. Alexander Fleming could discover the antibiotic penicillin, and it was first successful genetic engineering experiments were conducted much later, 1973. So right now you have so many drugs, so many vaccines that are still under trial, and then you have basic the most recent that they found out was some kind of medicine which were given has found has stopped your Alzheimer's disease in say in 6 months. So that is a very good progress, in fact, and it is going to be very useful also because it is because of protein misfolding that Alzheimer's takes place, or Alzheimer's is caused. So you get to see that papaya, soybeans, corns, all of them have been genetically modified also. So, and then you can see that environmental biotechnology is helping us try to reduce the pollution by producing or production of bioplastic. So, and then you have your oil spills that are being cleared up by your different kinds of microbes. So all this is like genetically engineered organisms that are actually doing that, and at the same time is production of bioplastic. It's in decreasing your plastic pollution. DNA fingerprinting, of course, has helped in different kinds of criminal cases also, and in case of forensic medicine. So when it comes to the industry part, you can produce your antibiotics and different kinds of enzymes, all of it. So in our country, we have BIRAC, which is Biotechnology Industry Research Assistance Council. It is known as BIRAC. And then DBT, Department of Biotechnology, Government of India. So both this BIRAC is a private sector, DBT is of course government. So you have large corporations, MNCs, all into this biotechnology industry, which is going to focus on health, medicine, food, agri-science. So what is it going to do? Increase skilled human, help in infrastructure, different kinds of policies and different kinds of factors, environmental factors. So when it comes to the different kinds of pioneers in biotechnology, you have Anton von Leeuwenhoek. He was the first person to discover bacteria. Gregor Johann Mendel was a, Austrian. Beside Germany, you have a country called Austria. Capital is Vienna. So he had conducted pea plants, pea experiments on the pea plants, that is Pisum sativum. So he's the father of genetics, and he was he was a priest actually. Louis Pasteur, he not tell about him. Uh, uh, he created the rabies vaccine, started or developed the pasteurization technique. And Robert Hooke was the first person to invent the compound microscopes. He saw the Koch cells as well. And then we need not tell about your Watson and Crick. They were given the credit for the discovery of the double helical structure of DNA using X-ray crystallography. It was actually another Rosalind Franklin's discovery, but credit was given to these two gentlemen. Cohen and Boyer, 1973, were the first to develop rDNA technology. And when it comes to the historical development also, from 1750 BC, the cheese was produced, yogurt, yeast, fermentation products. 1869, DNA was discovered in the trout sperm by German scientist, German Fischer. 1919, first time a Hungarian engineer called Karl Erki described or coined the term biotechnology. Of course, you have huge amount of research that went that way. By the 1970s, production of insulin, HGH, interferons, cancer, extensive studies started up. 1980s to 1919, again, rDNA technology boom was taking place, going on, in fact. And then you have plant tissue culture, so that you have genetically identified plants. By 1990s, again, we have the production of BT corn and soybeans, and gene from bacterium, that is BT. So your BT is your Bacillus thuringiensis. Corn, cotton, all of it. 1997, historically, dolly was the first animal to be cloned from your diploid somatic cells in Scotland. And then late 90s to early 2000, you can see that concerns about the stem cell research started to come up. And then in the 20th century, we have so many different kinds of biotechnological aids. The DNA sequencing, protein analysis, whatever you name it, we are all having a lot of biotechnology influence. Now we have something called as a nanotechnology. And then we have AI, that is artificial, cognitive and information sciences. So basically, when we have bioinformatics and artificial intelligence coming up, we are trying to see something else. Now, what is the scope of biotechnology? Again, it is a multidisciplinary pursuit. So you have different kinds of antibiotics, life-saving drugs, gene therapy, all of it coming under. You can use different kinds of drug testing, clinical testing, diagnostic tools, novel varieties of crops, all of it can be produced. Then you have production of food products, fertilizers, pesticides, beverages, and then of course wastewater treatment, irrigation, energy production, all of it. And then using of different kinds of synthetic enzymes that are also being produced, and we have to include alcoholic beverages. Antibiotics, you name your antibiotic, all of it has been produced only through biotechnology, and industrial production of the antibiotics is also has to be taken into consideration. Vaccines, steroids, vitamins, insulins, human growth hormone, pesticides, citric acid, all of it is produced. Now we are able to produce different kinds of skin, that is your artificial skin, different kinds of organs to the people who have problems. So when it comes to biotechnology and agri-science, we get to see that it is involved in plants, animals, environment, and agriculture or medicine. So what can be done to improve the quality of the plant? And in the animals, in vitro fertilization, artificial insemination, so that you get a better quality or better breed in the animal. Molecular engineering, you have the DNA mapping and sequencing. So you into yeast artificial chromosomes. So you can use that to help identify different diseases, find out a different medicine for the diseases. In the case of environmental science also, say production of bioplastic, production of genetically modified organisms so that they will be able to clear up the different kinds of pollution that is occurring, and increasing vitamin A or D enhanced foods, improve tolerance to different stress in, say, abiotic stress, or if there's any kind of saline tolerance. So the plants can be incorporated with such a gene so that they will be able to tolerate saline, high saline conditions. And if there is any kind of agricultural waste, it can be reduced. So this is one example. And then we have our immunoassay tests, your ELISA, Western blotting, and then your basic pregnancy test kit, your diabetes test or ovulation test kit. All these, are basically through the biotechnology only. You has that is to be seen. Okay. Example is the tire wash channel. In the case of health or agriculture, you can use different types of testing through biotechnological tools. Paternity testing is one. You can find out the ancestry of animals as well as. But what are the different kinds of problems that there is? Whenever there is a transfer of gene which is not necessary, though it is attached to a necessity, then it is going to have a problem. And ecosystems, if at all they are invaded with the different kinds of modified plants, then it is going to create a problem. If at all it develops a resistance against pesticides or herbicides, it's a problem because it's going to harm even the beneficial insects also. And then you see that the genetically modified organisms can have a different impact altogether along with the other organisms. So you can see the cost is going to be involved, so it is very expensive. Participation, accessibility is a problem. Not everywhere can you do all these biotechnology related experiments, especially in different kinds of these programs cannot be held so challenged or economically challenged countries, and then large countries are going to dominate and monopolize the demand and supply, obviously, and concerns over the kind of impact it has on humans. Will it produce allergens? Example is they have given starling corn and taco shells is not approved for human consumption, though it has been produced. And then is it like, what do you call it? What are the kind of concerns that we are going to face? And is it the awareness makes an important aspect? And then your genetically modified organisms are actually concerned right now. It's only that how much of it is aware, made aware to us. It's US, that's one of the single or largest producers so far of biotechnologically enhanced crops, basically, or GMO crops, and monosodium glutamate produced by the Japanese. It's known as your Ajinomoto. And then enzymes for animal feed supplements are produced. Your BT insecticide is going to produce cotton, different kinds. And then since the discovery of monoclonal antibodies, of course, it is used in diagnostic purposes, medicine purposes, especially for the treatment of cancer. All your cancer medicines are usually having monoclonal antibodies. So this is all about your and then when it comes to the enzymes also, you can produce using organisms different kinds of enzymes in bulk. So you have 4 phases: extraction, purification, concentration, stabilization, and integration. Again, these biotechnological programs or tools can be used in cosmetics, medicine, starch, detergent, all the other kinds of industry. Now we have a biocontrol agent which is Bacillus thuringiensis. It is a microbial insecticide, in fact, and it is neurotoxic towards mites, scorpions. And you have pheromones which help in pest control also, and crude plant extracts. So you have even your WAM fungi and plant growth via like Rhizobia fluorescens strains, and immunomodulators that are present in the vaccine industry, which play a role everywhere. Now, when it comes to genetic manipulation, also production of different kinds of vaccines, growth hormones, and then genetically engineered meat, which is basically not meat as such, but plant-based, so that there is a little bit less amount of killing of the animals. And then you have germplasm improvement and conservation through genetic diversity. So that is through hybridization techniques. Of course, your IPR, that is intellectual property rights, should be taken into care. You have to see about the right to information, and you should have proper policies. All this is taken care by by the government. So again, you should see that it impacts the anthropological, economical, social, all kinds of, and yes, ethics is very important again when we talk about. So these are the terms that we get to see: cloning, offspring, and this is all for today. And I would like to stop sharing and then see you tomorrow. Thank you so much.